Hello, I'm Dave Turr from UPMC, and today I'm going to be presenting our work on intramuscular nanofat injection to prevent muscle atrophy after peripheral nerve injury. It's well known that immediately following peripheral nerve injury, the process of muscle atrophy begins. And even complete nerve regeneration does not guarantee that the muscle will be reinnervated or will return to normal function. To put this in perspective, peripheral nerve injury affects up to 20 million Americans at a cost of over $150 billion annually. 41% of patients after peripheral nerve injury have not returned to work 1 year following the injury. Our lab has previously demonstrated that cultured adipose-derived stem cells have a protective effect against muscle atrophy after peripheral nerve injury, and that ASCs also upregulate pro-regenerative cytokines. However, cultured ASCs cannot be used clinically at this point. However, nanofat is a currently viable source of adipose-derived stem cells. Nanofat is obtained by mechanically processing lipoaspirate, and the shear stress caused during the processing causes adipocyte rupture and disaggregation of the stromal cells, including the ASCs. So in our study, 6 rats were sacked and their inguinal fat pads were harvested and processed into nanofat. We then studied 6 control and 6 nanofat injected rats, and I'll explain the exact process of that in the next slide. Then at 9 and 12 weeks post-op, we measured the force of contraction, histology of the retrieved muscles, and cytokine quantification from the muscles. So each rat served as their own control. The contralateral hind limb was left uninjured, and the experimental hind limb underwent transection of the sciatic nerve, followed by primary repair. And then in the intervention group, the gastrocnemius muscle was injected with nanofat. Again, at 9 and 12 weeks, the rats were anesthetized and the sciatic nerve was dissected to ensure healing and regeneration. Then the gastrocnemius muscle was isolated and a cuff electrode was placed on the sciatic nerve for stimulation. This allowed us to measure the force of contraction of the gastrocnemius muscle. In this figure, the gray bars represent the contralateral uninjured side and the black bars represent the injured side. Control rats are the ones that receive no nanofat, and nanofat designate the rats that receive nanofat injection. At both 9 and 12 weeks, we see that although there was a decrease in contractile force in both the control and nanofat groups compared to the uninjured side, the nanofat group had a significantly higher return of contractile force at each time point. Mason's trichrome staining also demonstrated that there was significantly more collagen deposition and therefore fibrosis in the non-nanofat injected group when compared to the nanofat injected rats. And finally, we also found significant increases in IL-1 beta, IL-18, and VEGF in the nanofat injected muscle, indicating an upregulation of pro-regenerative cytokines. So in summary, intramuscular nanofat injection at the time of peripheral nerve injury preserves muscle contractile force, increases pro-regenerative cytokines, and prevents muscle fibrosis. Our next steps are to expand the size of our study to a larger sample size as well as to a large animal model, and also to study the efficacy of intramuscular nanofat injection in humans after peripheral nerve injury. I appreciate your attention, and please direct any questions to email.